of the story about protein interaction site prediction. So again, the pairing will be on the same issue. Protein interactions for us are two proteins touch each other. What we showed is that there is a difference in terms of the frequency or the expectation for residues in interfaces between two different proteins, shown in red here, between two different parts of the same protein, shown here in yellow. An example for that is shown in here in yellow for this blue protein here. And inside of a protein here in green, what was yellow there is blue here, and the green is green. Again, what this shows is the surprise to which this type of amino acid is occurring in one of these classes, three classes. And you see that all bars deviate from the zero, and that the three classes somehow deviate from each other. There was an issue about statistical significance. We addressed that. We showed that there are even more different types that can be distinguished. We talked about this issue that statistical significance is not scientific significance. Briefly, statistical significance essentially is about quantitative difference. So if something is big enough in order so that a different data set or different sampling of the same data set would still show it as a difference, that is statistical difference, while scientific significance means that it has a scientific impact. So meaning you can do something with it. For instance, you can predict with this difference interaction sites, right? That is one example. And this ultimately goes back to a big discussion about correlation and causation. And since the year's beginning, so I made this point that predicting the sites from sequence alone, we can do it to some extent. What I show here is precision versus recall. And you may see this light blue here. This is as far as you can go in terms of predicting from sequence alone interaction sites. So there is a signal that is statistically significant and scientifically significant. You can use it for prediction, but the signal is not very strong. We need, in order to make this signal stronger, we need to put in, by the way, just in order to explain how we dial through the curve here, we simply use different, so these ones here are the strongest predictions, stronger predictions. So we have a neural network out with two output units. One is binding to another protein, one is not binding to another protein. Both of those would say binding to another protein, because in both cases this unit here is the majority, or the highest value, the maximum. But this is a much stronger prediction because the difference between the two is much higher here than here. And high difference gets you here in the curve, low difference gets you here in the curve. This is essentially how this curve is computed. Now, you always have to compare something such as this signal to what? To another method. Okay, if another method is not available, what do you have to compare yourself to? What is the minimal thing that you have to beat?
Random.
Yeah, so the coin, or the dumbest thing you can do. Now the dumbest thing you can do can be, in this particular case, is random. And random can typically be done in different ways. You can sample random in different ways. So you can swap the labels, for instance, is one way. When you compile random, you have to be careful that in computing random, it's a real random. A real random means that you sort of predict if there is an imbalance in your data set, that you pick up in random the same imbalance. Okay? The most extreme example, for instance, is you want to predict binding sites. 10% of the residues in a protein typically have binding sites. You want to predict them. The best prediction by a Q2 would be to always predict not binding, right? Because 90% of the data, 90% you would be right. And if you measure just how many of the ones you predict are right, if you always predict non-binding, obviously you get the highest Q2 much higher than random. Because if you chose random, you would get a level much, much lower than 90%, right? So you also have to compare what this random is beating. And that brings us to this next issue. Random is not always just random. It could also be something that you, understanding the problem a little bit, do better. So for instance, at some point when you optimize the feature space that you use, you could call one-hot encoding the background, because that's the simplest you can do. And then you would sort of, instead of comparing yourself to random, you would compare yourself to one hot encoding. This is another way, because it's the dumbest, or whatever is the dumbest way in which you can solve the problem. This is not necessarily random. And you clearly want to beat that, right? And sometimes this is, for students in our group, this is sort of a fun exercise just before you publish, to come up with something that is dumb, but actually comes close to your own method. And sometimes this causes pain when you get too close with a dumb thing. Anyway, so the big jump I explained to you is by introducing evolutionary information. Evolutionary information is simply, you take the sequence, the protein sequence, you find a bunch of proteins that are related, sequences that are related. Related means most likely they evolved to do the same thing. If they evolved to do the same thing, we assume that in the pattern of which residue has always D, which residue has, this is not a good example, but in this particular case you have a D here, but in the others you don't have a D. Right? So where are residues necessarily conserved, meaning the same in this entire family, where not? This signal is put into the network. When you do that, you get a much better prediction. So this is sequence only, and that is sort of using the evolutionary information and a little bit more than that. I also talked about the ones up here, sort of are hotspots, and you can use this to predict hotspots. That gets us to the next level. The next level really is, so far what we did is we took a sequence and predicted whether or not a particular residue binds another protein. But I did not talk about what other protein, and that's what we're going to talk today. Now, let's get back into some sort of generic statements about machine learning or artificial intelligence, generic issues. So, arguably, you could argue there is sort of the old world in which a computer beats a chess champion, Garry Kasparov, here in '97 for the first time. Deep Blue beat him, and the new world in which Go, and for the game Go, a champion is beat. What's the difference between these two worlds? Yeah?
I think the first one is that it's very brute force. It takes every possibility into consideration, and this is more pragmatic.
Exactly. But for me this is important. I make this point not because these two games, one is done by brute force and one is done by machine learning or artificial intelligence, as it is now called. The difference here in time for me is so important because underlying is something else. These are times when computers were the machines that operate because they are brute force. They can do simple operation very quickly. And that's the way we have thought about computers for most of the time. Even when you guys went to school, this is the way. Even when you started to study, you thought about computers as big muscle machines. This is over, right? Over the last 2 or 3 years, this complete idea about how we think about computers is changing. Computers are no longer these muscle machines. Computers are things that use artificial intelligence, that use software to do something completely different. Today, if you run chess on your laptop, you use artificial intelligence. The laptop could do most likely what Deep Blue did 20 years ago, right? The power is not the issue. The issue is the artificial intelligence program on your machine is so much simpler, or so much lighter. It needs much less space, much less CPU. It can just run in the background and you can do whatever you need to do anyway, right? And this is because we now use or change the way we use the computers. We reprogram them. We no longer use brute force methods, but we use sort of approximations from artificial intelligence. For instance, the Kramer's group upstairs, they are sort of making quadcopters fly autonomously. And a lot of these algorithms initially to make things fly in the world are brute force. So you completely understand the algorithm of how to find out certain parts of the environment. They no longer do that, simply because the little quadcopter is a small ship and artificial intelligence fits on less space. So the difference between these two is much bigger than we can now handle Go, and previously we could handle chess. The difference is it's a shift in the philosophy of the way we perceive computers, right? This is what we have to understand. And underneath is sort of what today again is called artificial intelligence. The term has changed, and it is. The idea often has been that this machine is some sort of dark thing and that the understanding of the world is hidden in that machine. And that's a complete misunderstanding. a lot, you know, understandably AI is one of those buzzwords of the day, so people are trying to more and more understand what the AI actually learns, and this is clearly part of it. But there's another aspect of it, and that other aspect is you can actually get the rule out of the black box. Now, when you get the rule out of the black box, people look at the rule and they say, for instance, in this example of a machine that predicts which residue binds another protein, you find that, okay, if that ALA is an alanine, and if it is conserved to 22%, and if it sits next to something that is to 2% helix, and then it it goes on, and then you read through the sentence, and you realize, oops, that is not a meaningful rule, right? So why is it not a meaningful rule? Ultimately, what I'm posing here is the black boxes are applied to complex problems that historically, again, this is changing. I said now they apply to problems that we have already solved. But for many problems still, artificial intelligence is applied today too, are things we cannot write down an equation, okay? We cannot write down simple rules. And when we extract the rules from the system, we are surprised that they are complex. They are as complex as the problem, right? And that is what people sort of don't understand or forget. It matches the complexity of the problem. That means that the understanding the machine gets is there. It's just I'm not able to handle it. It's not that it's a black box, okay? But part of that I completely understand. Part of that is the challenge to get really the rules out, to understand what the AI is doing, and that is really an interesting challenge of the time. Now, in order for all of that to work, there are two sort of main ingredients: the cross-validation and the representativeness of the data set. Now, representative of the data set, essentially you're trying to do something to predict some future. And the data you have must be representative for that future, otherwise it doesn't work, right? It's a trivial statement, but to make sure that this is the case, highly non-trivial. And in many cases things go wrong because that in fact is violated. This issue of cross-validation, ultimately cross-validation is a big word. What I mean ultimately here is the assessment of how well your machine works. It is better that you have a machine that predicts binding sites with 95% than with 75. That's true. But as long as you know the performance of your method, you most likely can live with it. So the worst thing that can happen to you is you have something that predicts 75 and you believe is 95. That's the correct assessment. And that correct assessment, typically the other thing is, if you don't get that right, that means you don't understand your problem. That means that you're actually not extracting some truth out of it. You're not extracting a complex rule. You're sort of simplifying the issue by making a mistake. Again, cross-validation is about hiding data, and the way this is done is highly non-trivial. It is trivial for the methods that you typically use. It is trivial the way it's described, but in reality things do not work out in the same way as they seem to. It begins by issues that you cannot choose your patterns at random, because there's, in proteins, you know, there are families, right? You have to find out whether in your data there are relations that are trivial, and you have to group them. So that's the first sort of pre-processing step. For every data set, this pre-processing step will be different. And in many cases, you will encounter data sets where nobody who has ever worked on that has any idea about the noise or the bias that is in those data. This is highly non-trivial, but it's a very, very important issue. I told you at some point that using one split of training and testing is not enough. You have to rotate through. I said that one split in terms of train test or train validation is not enough. You need a three-way split. You need a set that you use for development. You need a set that you use for hyperparameter and model choice, or hyperparameter optimization, or more generically model choice. Hyperparameter optimization is nothing else than model choice at the end of the day. But model choice as a word also includes, are you going to use, what type of neural network are you going to use? Are you going to use an SVM instead? How are you going to combine different prediction methods? All of that is part of method choice, model choice, sorry. And all of that has to sort of be assessed in cross-training. And once you have the final system that you want to publish, that's the moment when you test. And you never test two different solutions. You never test two different alternatives. You test yourself compared to something that somebody else has developed. But if you sort of choose the best, and I'll get maybe back to that in the question and answer sessions, But you can never choose a model based on the test set. The test set is only relevant to assess how well you do, not what is best. Then when you do all of that, then you maybe find some new data set. And in the new data set you see that all your estimates are consistent wherever you do. If you do that, I call it pre-release data set, then you may get to a point where you publish a paper, and this paper gets a lot of citations, as this particular one. And not only, but in fact its estimate of performance is actually surviving for 20 years. This is a rare event. So I was involved in this first study, as you may have recognized from the photo. And I did the same thing again in another paper here a few years later, and that totally was wrong. So there the estimate was absolutely wrong. And in fact, again, we published a few years later a paper that said this is absolutely wrong. And the reason was that there was some issue with the data. We could have possibly foreseen that. Possibly we could say, well, we don't know, we cannot check the experimental accuracy, and if I have an overestimate, then it's not my mistake. But in some sense, this is not as simple as that. Ultimately, it all boils down to we made a mistake. And the mistake had led to the situation that we incorrectly estimated how well we understood reality, what the machine learning did, in fact. But anyway, if we do that correctly, we can gain understanding. And in fact, the prediction is the acid test for being able to understand. I showed you an extreme example here, where from the prediction of the hotspots, that's a prediction that a model does for which I only put in that two residues are close to each other. I never put in the label hotspots. Nevertheless, the system extracted knowledge that allows me to predict hotspots. So that means we have an example where the machine learning extracts truth that is never put into by me. It's somewhere in the data, right? But I never trained hotspots, okay? And nevertheless it can predict hotspots. That's an example where you learn that the system has learned something about the underlying biophysics of the way proteins bind, okay? I believe you're right. I forgot to switch that on. Oh no. Anyway, so this is an example where that works. So here, how well hotspots are predicted, I showed you that image before. So we can gather understanding about data, understanding that really is beyond of what we actually know, what we actually put in. And it can go very well, but it can go very, very well. Very wrong. And an example is for the topic of today, where we predict now, I want to predict that this protein binds to that protein, okay, and not to some other protein. And in the first instance of the game, I'm not predicting where it binds, I'm just saying A binds B, or A does not bind B, all right? So a very binary thing. And in order to do this, so we tried to do it with a very simple idea, where we have a neural network into which we put in this sequence of one protein and the sequence of another protein, and then we essentially say they bind. And then we sort of move these windows through the entire protein. That completely failed. And we realized we have to sort of fix a bunch of problems in order to solve this problem. And this, in fact, to go through this slide, and I'm not going to go through this slide, but essentially this is a set of methods that had been developed in my group over more than a decade. And we needed all of that to come to the point where we can essentially say, now we can predict from sequence alone. Now we can do something like that. And in the first step, what we can do is we can simply say A binds B, not where. So we're not really shifting the window through. We're using something else I'll get back to later. But, so this, so the first but, there's two buts through the story. Number one, method number 41. So this is the method that took we had a long development time here. But method 41 was very, very, very good. Method 41, although it was very, very good, had a tiny bit of a problem. So the problem was, what we did is we used proteins from the PDB. So you in the room by now, you know the PDB are protein data bank. This is the database of database of proteins for which we know the three-dimensional structure. Okay. So when we wanted to say that something is a positive case for protein A binds B, We looked it up in a database where we know in real detail how two proteins bind. We know that A binds B from this database PDB, and we know exactly where they bind. Okay? That's our database of positives. Then there's our database of negatives. In the database of negatives, we simply looked at, and another issue, by the way, we can do this only for one organism at a time. So in this particular case, I'm going to show you data for human protein prediction. So in the negatives, we take all the proteins that nobody has ever observed to bind. Not only are they not in the PDB, but also by sort of low resolution experiments. So PDB, determining where they bind, the three-dimensional structure is very expensive. Determining that two proteins bind at all, in principle, is much, much cheaper. Not as in principle, is much cheaper. There are much cheaper methods out there, so there's much more data of low resolution that protein A sort of, you can argue, may bind B, or at least it's data that they do bind. It's not good enough for us to use for the positives, we argued, but it's certainly good enough to say that if nobody ever observed that two proteins bind, then we call them, they don't bind. Okay, there are two issues with this. What are they? The one statistical difference between the proteins in the PDB and the proteins in SwissProt is they are on average longer. But this is not the feature. In some cases you can do machine learning. So this is one feature that the simplest feature of the machine learning got, right? But it turns out for many proteins this is not the feature, because many proteins have the same length in both. And for those there is no principal difference that we had been aware of. But the machine learning simply found that there in fact are differences. Okay, surprising. And ultimately that means that if we wanted to talk about something else, that is cool, because now we can possibly predict whether a protein whether we can do a structure for a particular protein. This is sort of a side effect of that. Absolutely not wanted, a total coincidence, and we never pursued that. There is some difference. We don't quite know what it means. It's a longer story how to pursue it. In the context of what I'm talking about now, this is just nonsense. It's just something that happened because you sampled the positives, one set of your data, from a different distribution than the negatives. And that is a no-no. Now in this particular case it's a little bit more tricky how to fix that issue. Because we cannot take everything that is not observed. So here essentially I make I take this assumption everything that nobody has ever observed is negative. I could not do that in the PDB, because the PDB is a tiny subset. Not all human proteins have been studied in the PDB, right? So there I cannot just stay in the PDB and flip this. Impossible. Look at the red in the PDB, that's impossible. So I have to, one way, ultimately the way we solve this here, or we address this, is we sort of pick the ones from here that are in there. So we could pick the label from here, but we took the sequence from here. And one other thing, answering to your question, Stanislav, is when you determine a structure, typically you shorten it. That's why those are shorter. And shorten it, typically what you try to do is you throw things out that elude structure determination. And that makes them shorter, so they are difficult to get a structure for. So what is difficult to get a structure for? For a structure you need to put the protein into a crystal. If you have sort of floppy elements that sort of stand out somewhere, cut them out, because they are most likely in the way of getting a good crystal. So cut out anything that sort of is not round and crystal forming. But then when we get back to the sequence of SwissProt, we can take the entire thing, including that thing that is cut out, right? So now the positives and negatives are sampled in terms of sequence from the same database. And that sort of gets around this particular problem. Now, the second major issue really is the dataset preparation. And the dataset preparation issue, we already discussed this issue here. If we have three methods, the performance difference being that, one question that you may ask, do they differ? For instance, in this particular case, you may say method 3 is the best and performs at 37%, and the question is that correct? And there's a bunch of questions you have to ask. You have to, for instance, ask, are these numbers all compared to the same data set? Are these numbers, in fact, I say performance, is it actually really the same performance measure? You very often have papers that use different ways of defining even AUC. Just look at the detail of the method section. You will be surprised how often you see details differing and making a big difference. And then you have to look at statistical significance, so you have to look at error levels. Now, when you look at error levels, then I'm going to skip over that. Then you have to can see whether they really differ. But again, so the point. Let's get back to the issue we want to get at. We want to sort of predict pairing. We have to do some filtering. But one thing that we absolutely have to do is we have to reduce redundancy. And that is back to what I said before. In every data set there's some bias, and the decision what is considered bias depends on what you want to do with the data. Okay? In our world of proteins, we know we have to group them. Okay, we have to group them into families and then say we can only take one representative from a family in a test set, and we can only take this family in training. If we take this in training, we cannot take another representative for testing. Things like that, right? That's part of these issues of grouping. Now, for protein interaction prediction, the story gets slightly more complicated, because now you're not having a grouping of a family. You actually have an interaction that you have to group. So I'm sort of indicating this by now you have two halves of a circle. You have the group of one half and the group of another half. And you have to somehow handle the issue that they interact and that you have to group on this issue of interaction. So for instance, and that's Yonki Park and Burkhard found that out first, they looked at 3 different cases. They looked at cases where we have a protein A and B interacting, and essentially you train on A and B and test on something else. So in the first case, both proteins A and B. So you have examples. Let me illustrate that. You have a protein A and B, and you want to test its interaction. So case 1 is that A has been used for training, but say A with C. And B has been used for, so this is case 1, and B has been used for training, but B with D. So in your training set, you have cases of A and you have cases of B, not interacting with A and B, but with other proteins. Okay? And in principle, you would argue, this is not bias, because whether A interacts with B or A interacts with a completely different protein C, it's important to know, it's important to distinguish, right? Why should you not train on A and C and on B and D when you test on AB? As long as there is sort of some difference between all of, say all of these proteins, 4 proteins that I have, are unrelated in terms of a family. Like, this should be okay. You may argue. And case number 2 is here where either of these 2 has been used another one. Say you had in your training A with C, not B with D. Okay? And in the case 3, you're not allowing any of the 2. So when you test on A and B, neither A nor B have ever been used in the training. Okay? And what they showed is that, as already indicated here, this leads, the more case 3 is predicted much less well. Performance is much worse for the proteins that have never been used for training. Here is the same story shown with a little bit more detail in terms of recall versus precision. Let's not get into the detail. The issue is that in order to do protein interaction predictions, you have to distinguish these 3 cases. And all 3 cases may be relevant for your method, but very often what you want is a method that really predicts new proteins that it has never seen before, right? And that means that the way you sort of pre-process the positives and negatives is very important. Now, here's the next issue. Essentially, the way we do this here is we say the positives are the ones for which we have an observation, and the negatives are the ones for which we have no observation. And again, as we discussed, many of the negatives are wrong. In fact, we're building this method to find the ones in the negatives that are wrong in this construction, right? Now, we have this other issue that you brought up already, which is that the balance between negatives and positives is an issue. We cannot have them balanced. That's very clear. In nature, if I said that a protein in human interacts on average with 100 others, it has 20,000 it could interact with. So the vast majority of proteins it will not interact with. So the negatives, if I do an all against all, 20,000 by 20,000, the vast majority of those will not interact, right? So I will clearly in nature have a completely imbalanced data set. But how imbalanced is it going to be? What is clear is I have to much oversample negatives. So the set of negatives has to be orders of magnitude larger than the positive set. Is that clear to everybody? Any question about this statement? Why? I assume it's clear to everybody. Now, still this question remains the case. The negatives in some sense are the ones created in silico, if you want. I mean, at least not quite. We have no evidence of any data that they interact. It's not quite in silico, but it's a data set that is very similar to one that you would, if you predicted that every pair of proteins doesn't bind, this would be much simpler, much closer to the data set that you actually create. So, should we actually apply bias reduction on the negatives, or is it good enough to do redundancy reduction on the positives? So we found out that we had to take the negatives and sample them. What we essentially see here is a deviation from the correct solution, and the correct solution is deviated from if we do not cluster the negatives. Why? And that is what you're trying to answer now. Ultimately, why? The answer is because we are oversampling some parts and it finds similarities. Essentially, again, it sort of groups things here. It finds these groups and looks at these groups to make predictions. And that's irrelevant. So it picks out simple features. Now, this means that in order to do cross-validation, and I'm not going to go through this, to sort of decide which pairs of proteins can be in the positives, negatives, which can be in training, testing, in cross-validation sets, is becoming highly complicated. So you essentially have to select on networks, and you have to be very careful in which way you do the selection, and that makes it complicated. The next challenge for any artificial intelligence is always the way we get the data. And the major thing here really is this issue: we need as much data as possible. We need a lot of data, but we don't really have a lot of data. I mean, we have, at this point, for human, what we have, I said that maybe every protein binds to 50 other proteins. Currently, good data we have for maybe 5. So we're at least a factor of 10 away from the entire thing. So it may be way too small. We don't know. That brings us to the question: how do I define? How do I find out? What is enough data? What can I do? You want to take less data. So you have your data set, you halve it, and say your estimated performance for the entire data set and for the half data set are very similar. What is it that you learn? So you look at 2, the gradient in this particular case being on simply 2 points. So you have data set size here. This is the data set 1, and this is the data set 1/2, and this is the performance, and say this is the performance here, and for 1/2 it's here. And say the difference here is the delta is essentially in the ballpark of the error, or the sigma. It's in the ballpark of a standard error. So you can hardly distinguish these two numbers. Statistically, you cannot distinguish these two numbers. What does it mean? Now, you were one of the people who understood it in the first place. But go ahead.
Yeah, for me, it means that if we take more data, we already have enough, so it doesn't improve.
So that would then bring us to this challenging statement, or the statement that half the data was enough. Well, if half the data is enough, if half is giving the saturation point, then the data I have is enough. Okay? Is that right? In particular, assume that this performance level, say we are looking at precision, and just for the sake of the argument, say this is 0.1. We have 10% precision. So it's nothing like, you have a method that's completely right. 90% of the time is wrong. So it's it is way, way, way better than random. Because again, 1 in 20,000 is a random, or something like that, right? So the random is very, very low. So we are many times above random. But anyway, so what could be the other problem? Yeah?
The wrong model.
Exactly. So it could simply be that the way I use the input. So the wrong model has many implications, right? But one way of saying wrong model is simply, I do not use all the input information that I need. I can, from the input information that I have, I just hit the ceiling. And maybe I would have hit the ceiling already with 1/10 of the data, or 1/100th of the data. Essentially, I need one protein to see that PDB proteins are longer, or something like that, yes?
But it can also mean that from the data you have, it's principally impossible to derive the measure you like.
That's the next point. That's another very, very important point. And these are, in principle, completely uncorrelated, unrelated things. The model could be completely wrong, or the data could be completely wrong. This is essentially my two main points about cross-validation and data. Yes? I interrupted you, Stanislav.
No, like the question is, how do we find out whether they're so uncorrelated that you cannot derive any information from them? Like, how do you find out that this is the case? Because that's kind of like a lot of lost work. It's correct.
Ultimately, again, so part of that is answered by the precise value here. If that value would be 100, your question would be easily answered, right? In a case where it's so far away from 100, this is very, very challenging to find out. Not easy. But let's just say that at least we would assume from this here, we would assume, we would try to, if it is the model we can address. We can try different models, okay? And for all models we still see this. It could still be the model, but we would begin to argue, well, maybe it's data set size. How could you continue? Well, the next thing you would do is even smaller and see, is there a point at which this goes down? Is there a point where the data set really is so small that at least that is not enough? If you cannot find that thing, then it is either the data or the model, and you don't know how to fix the model. If there is somehow increase somewhere, maybe not at the point of half, but somewhere, then at least you believe you're in the right direction. If, however, the result would have been completely different, say for half the data set size, you get half the performance or something like that. What is the conclusion then? Absolutely. That is exactly, so here we have a typical example where we know, we need absolutely more data. We can maybe not get it. I don't know what that again means for my model. Maybe more data, or my model is too big. It could also mean that. The way I size my model is that my model needs more data. Maybe at that point you would sort of first tune down the model and see whether with a smaller model you sort of still get this maybe the smaller model produces something like that. Or maybe the smaller model produces something like this. Or something like, sorry, that's the wrong point. Where the difference between the size distribution is sort of shrinking. And then you believe so. Then you sort of realize maybe it has to do more with the model than with the data, or also at least with the model size. Is it related to the data? You would still not see, but you would get a clearer signal. So the first thing is, if you have your data, play around by just cutting it, doubling it, to just see what is the effect. It has some implications on what you're going to do next. You're not completely getting the answer, but you, in some cases, do get a clear answer of what is wrong. You may just not be able to fix it. Now, ultimately, these cases here, the one case whether the A was in the training or not, is something about more data. Because if I kick out all the cases where I've ever used A and B, I have way fewer interactions. Right? If I can train on all the proteins where A binds C, but B binds D in order to predict A and B, I have much more I have many more cases than if I have to kick out all the proteins that I've ever seen. Because ultimately in this training, if you think this through, any protein pair that you predict, you could have never used in training any of the two, which is a tremendous reduction, right? But anyway, the next issue, and I'm just skipping over this slide. The next issue that we had is, what if we had the same example here, but in a different context? Let's just look at the case 3. So I never train on proteins that I test on, okay? But even if I don't do that, I still have the question, okay, should I have an example for A binds C and A binds D. Should I train on both? If D and C are very different proteins. Or is redundancy reduction meaning that I use A only once? And it turned out for us, the moment we sort of had some cases where we had more than one interaction, so in some cases we had A binds C and D, in other cases we had B binds only F. So we had some proteins that have only one partner and some proteins that have more partners, and immediately the neural network extracted exactly that information. It simply distinguished between binders and more likely binders and less likely binders. That means we had to throw away all instances or repeated instances of A. We could use A only once. And that same was essentially true for the negatives, except for the sampling issue. So negatives were sampled, I believe we sampled negatives 50 times more. So we had A 50 times as a negative, but not more than 50 times as a negative. That was the challenge. The next challenge came. We are now having a data set that has relatively a few thousand protein pairs. This was pre-deep learning, but even with deep learning now, we have still not reproduced the results that we created before, simply because we have so few examples. We believe maybe we haven't done it right yet. But we still have not controlled this problem here with deep learning. Controlled, I mean a method that is better than what we have without deep learning. That's my definition. So how would you solve the problem? So you want to predict kernel. So the same machinery, the K-mer machinery that we used for predicting subset localization, you can use here too. Again, the profile kernel SVMs that we used, the idea is you look at K-mers that are conserved within families of related proteins. So if you think about this, what it means is you're looking for two proteins that have a lot of conserved K-mers that are corresponding to the training set. So essentially you're not predicting based on sequence alone, you're not predicting based on biophysical features, you're predicting sort of on matching K-mers between these pair, right? Now, in terms, and then when you do that, you sort of get a method that is in blue here substantially better, and in both blues here substantially better than the best methods that were available at the time. And I'm not sure whether it's still the best. At the publication a few years ago, that clearly was. The difference between the dark blue and the light blue is one about using some experimental data. That is the dark blue. So using some additional experimental data, here expression arrays helped you to do a little bit better. No, the other way around. The expression arrays did, in fact, for the problem here, where you predict for things where one protein was in the data set before, you in fact improve by using expression. For the case where you have completely new predictions, it actually hinders or is bad for you. But anyway, so the point now is, this. I hope you remember how this SVM profile kernel works. If not, get fit before the exam to sort of remember it. The point that I want to make here, essentially it looks at K-mers. The length of these K-mers in this particular case is 5 mers, so 5 consecutive amino acids. How many pairs do you believe are relevant for making these decisions? So we have all possible k-mers in a protein. That's a large number, right? We have two proteins, and ultimately what we looked at is how many, say 50% of the information for making a decision, for the performance, right? For reaching the performance P, 50% is contained in n k-mers, and it turns out the other 50% is contained in the long Gaussian tail. And my question is, so how many go to, say, the Gaussian tail of 70%? So 70% of the decision, how many k-mers do you need for that? Ballpark. And the surprising answer is hundreds of thousands. So we're building SVMs here with over 100,000 input parameters. So these are huge devices, actually. And we have just a few thousand samples to train this on. So it's sort of an edgy issue.